The nervous system is a major player in maintaining homeostasis. Sensory neurons monitor internal variables, such as blood pressure and the plasma glucose levels. The set point for many essential variables, such as blood oxygen and the osmolarity of our extracellular fluids, is located in the brain. The brain is also a major command center telling effectors, such as muscles, to contract. Finally, the nervous system monitors the effects of its own behaviors, so we don't overshoot or undershoot the set point. We call this continuous monitoring of the effects of our behaviors and the ensuing adjustments feedback. Let's review the structure of the nervous system and see how it contributes to the maintenance of homeostasis. The nervous system is divided into the central nervous system and the peripheral nervous system. The central nervous system is located within the dorsal cavity of the body and includes the brain and the spinal cord. The peripheral nervous system originates with the 12 pairs of cranial nerves off the brain and the 31 pairs of nerves off the spinal cord. The nervous system allows for rapid communication within the body and also rapid movement of the body. The fact that animals have nervous systems is why they are called animals. They're so animated. Among the many functions of the nervous system is a sensory function. The nervous system monitors conditions inside the body, like the pH of our blood. It also monitors conditions on the surface of the body, such as a bug crawling on your arm. It also allows us to monitor our distant environment. Our eyes and ears are our primary distance receptors. They allow us to anticipate bodily changes before they occur so we can avoid them. If you step blindly into the street, you might get flattened by an 18-wheeler, and all the homeostatic mechanisms of your body won't be able to pull you back together again. Instead, we use our distance receptors to look both ways, and only when the coast is clear do we step into the street. Our distance receptors are important sensory elements of our feed-forward reactions. The nervous system, primarily the brain, permits us to store our experiences so we can use them in the future as a guide to more successful behaviors. Without the memory function of our nervous system, we would not be able to learn anything. We would keep making the same mistakes. We wouldn't even recognize ourselves when we looked in a mirror. The integration function is the ability of the brain to bring past experiences to bear on current experiences so we can make better decisions. This is sometimes known as the executive function of the brain. The brain is like an executive in an organization, receiving reports from different parts of the organization, combining them with past experiences, and then deciding how to respond. Let's say you found yourself in the road and a semi was bearing down on you. An ability to see and hear the oncoming semi would not be enough. The ability to recall a lesson from a movie in which a guy in the street got killed by a semi would not be enough to save you unless you had a motor function. By motor function, I mean the capacity of your nervous system to control effectors, like skeletal muscles, so you can escape from danger. Finally, we have the conduction function. The nervous system conducts all parts of our body, so if you step on a tack, your cell phone may fly from your hand into the air. You may shout an obscenity and then reach down to pull the nail out of your foot. All this can happen within 5 seconds thanks to the conduction function of our nervous system. The primary cellular building blocks of the nervous system are called neurons. Some neurons are the longest cells in the body. I have neurons that extend all the way from my foot to my spinal cord, several feet long. There are hundreds of different types of neurons, but for simplicity we will reduce this to three major functional types: sensory, motor, and interneurons. All three are shown here. By definition, sensory neurons conduct signals toward the central nervous system. Motor neurons conduct signals away from the central nervous system toward effectors like muscles. Interneurons, also called association neurons, link sensory to motor neurons. In this cross-section through the spinal cord, you see one interneuron. But this is among the simplest of circuits. Your brain is one large mass of interneurons. If you're withdrawing from a painful stimulus, you want a quick response with few interneurons. If you want to pass your physiology exam, you'll need billions of interneurons that compose your brain. Here you see the two types of peripheral neurons, motor and sensory. Here is the cell body of the motor neuron. The blue part is the nucleus of the cell. Branching off the cell body are dendrites. Dendrite is from a Greek word that means tree. On Dendrology Day, you hug a tree. Don't the dendrites look like the branches of a tree? The dendrites are the antenna of the cell. They monitor what is going on outside the cell and then pass the information on to the cell body. The cell body then passes the information to this long process called the axon. The axon conducts a signal to effectors, usually muscles or glands. Note the direction of conduction is from the dendrites to cell body to axon. The other cell is a sensory neuron. Here is the cell body of the sensory neuron, and inside, in blue, we see the nucleus. Note that branching off the cell body is a single axon that splits into two branches. The branch that leads to the CNS is called the central axon. The branch that leads towards the periphery is called the peripheral axon. At the end of the peripheral axon, we find dendrites. Note that dendrites are off the cell body on the motor neuron, but they're off the peripheral axon on the sensory neuron. In both cases, they act as cellular antennae. In both sensory and motor neurons, the usual direction of conduction is from the dendritic end towards the axonal terminals. Let's have a quiz. Would you say this neuron is a sensory or a motor neuron? Note that the dendrites are off the cell body, and that the axon is unbranched. So this cell resembles a motor neuron. Note also that wrapped around the axon are support cells called glial cells or neuroglia. They're shown here in green. They look like a chain of hot dogs wrapped around the axon. The glial cells that are wrapped around the axon of this motor neuron form what is called the myelin sheath. It's a cellular lipid coat that helps to insulate one axon from another, like the insulation around an electrical wire. And it also acts to speed conduction of the signal. Myelinated neurons conduct signals faster than similar ones that are not myelinated. Usually, long axons are myelinated. If you've dissected a brain, you know the brain is made of white matter and gray matter. The white matter is due to myelinated axons. A neuron is an individual nerve cell. A nerve is a bundle of axons. The relationship of a neuron to a nerve is the same as a wire to a cable. Here we have a cross-section through a nerve, and inside you can see many axons of nerve cells. Here's an axon, here's an axon, here's an axon, another axon, another axon, and here's an axon that's been pulled out. And you can see the glial cells wrapped around that axon, forming the myelin sheath. The function again of these glial cells is to insulate one neuron from another inside these large bundles that we call the nerves. Individual nerve cells are too small to be seen with the unaided eye, but bundles of axons can be seen and named, such as the sciatic nerve, ulnar nerve, optic nerve, and so forth. Neurons are not loners. They work as a team called a neural circuit. Here is a simple neural circuit. It's composed of a sensory neuron, an interneuron, and a motor neuron. Let's begin our analysis with the sensory neuron. This is the dendritic end of the sensory neuron, out here in the skin. Recall the dendrites are like the antenna of the cell. So the neuron is monitoring conditions going on out here in the skin and can generate a signal that passes into the peripheral axon, past the cell body, into the central axon, and on into the central nervous system. There, the signal will be passed to the interneuron at its cell body or dendrites. The interneuron passes the signal along its axon to the motor neuron. The signal is passed to the dendrites or cell body of the motor neuron, which then conducts the signal along its axon toward the effectors. Here, the effectors are skeletal muscle cells. Another quiz for you. Is this a sensory or motor neuron? It looks more like a motor neuron because the dendrites are off the cell body and we have a single axon process. Note the axon is also myelinated. A stimulus occurs at the dendritic end of this neuron, which generates an electrical signal proportional to the strength of the stimulus. If this electrical signal is strong enough, it will generate an action potential along the axon that passes from one end of the axon to the other. Let's look at the nature of this electrical signal we call the action potential. Here we're comparing the intracellular fluid of the neuron, shown in yellow, with the extracellular fluid, shown in blue. To understand the nature of the action potential, you need to recognize two forces at work across the nerve cell membrane. Recall the intracellular fluid has a different composition than the extracellular fluid. For example, sodium ions are more concentrated outside the cell than they are inside the cell. Meanwhile, potassium ions are more concentrated inside the cell than outside the cell. Sodium and potassium ions are both positively charged, monovalent cations. Inside the cell, there is a predominance of negatively charged ions, including nucleotides, phosphates, sulfates, and proteins. Outside the cell, there's a slight excess of positively charged ions. As a result, there are two forces acting across the nerve cell membrane: a concentration force and an electrical force. The concentration force is a difference in concentration of a solute on either side of the membrane that tends to drive the solute through the membrane down its concentration gradient. The second force is due to a difference in charge across the membrane. If you place the microelectrodes of a voltmeter just inside and just outside the plasma membrane of an axon, we would see that there is a difference in charge of about minus 70 millivolts, with the outside positive and the inside negative. This difference in charge can affect the movement of ions across the membrane. Remember, like charges repel, unlike charges attract. For sodium ions, there is a concentration force directing it into the cell. But because sodium is positively charged and the inside of the cell is negatively charged, there's also an electric force trying to drive sodium into the cell. Two forces are trying to drive sodium into the cell, but it does not go. Why not? The membrane is normally impermeable to sodium ions. Sodium is beating on the membrane saying, "Let me in, let me in." But there are no open doors through which sodium can enter. The potassium story is a bit different. Potassium is more concentrated inside the cell than outside the cell. However, because the outside of the cell is positively charged, and potassium ions are positively charged, the electric force opposes the concentration force for potassium, and potassium stays put inside the cell. Let's review. The green symbolizes intracellular fluid. The white stands for extracellular fluid. There are two forces trying to drive sodium ions into the cell: the concentration force, shown in blue, and the electric force, shown in red. But sodium ions don't move because the membrane won't allow it. Meanwhile, the two forces acting on potassium are acting in opposite directions. The concentration force is trying to move potassium out of the cell, but the electric force is trying to keep potassium in the cell. The two forces largely cancel each other out, so potassium stays inside the cell. Now we're going to create a disturbance in the membrane that will trigger the opening of gated sodium channels. Sodium will rush down its electrochemical gradient into the cell, taking its positive charge with it. And as a result of sodium entering the cell, the electric force across the membrane changes from minus 70 millivolts to minus 60, minus 50, minus 40, minus 30, minus 20, minus 10, and finally, as sodium continues to enter the cell, the electric force goes to zero, meaning at that place along the membrane, there is no more difference in charge across the membrane. Nevertheless, sodium continues to enter the cell. What could be driving sodium into the cell if there's no electric force? The only other force is the concentration force. There is still a concentration gradient for sodium, and so sodium continues to enter the cell, taking its positive charge with it. And as a result, the electric force re-emerges, but this time opposite to what it was before. Now the membrane becomes plus 10 millivolts, plus 20 millivolts, plus 30 millivolts, as positively charged sodium ions continue to enter the cell, effectively reversing the electric force at that location. At about plus 30 millivolts, the voltage-sensitive sodium channels close so that no more sodium can enter the cell. But what has happened is that we have reversed the electric force at that location in the membrane. Next, what happens is potassium channels open up. And as a result, potassium ions flood out of the cell. Why? Number one, because there is a concentration gradient for potassium, directing potassium to the outside of the cell. But now there's also an electric gradient driving potassium out of the cell. That electric gradient was generated by all those sodium ions entering the cell and reversing the electric force. So when the potassium channels open, there are two forces driving potassium out of the cell. And as potassium leaves the cell, it takes its positive charge with it. And what happens is the electric force reverses again. We go from plus 30 to plus 20 to plus 10 to 0, meaning once again there's no more electric force at that location along the membrane. And yet, potassium ions continue to leave the cell. What could be driving them out of the cell? It must be the concentration force. So potassium continues to flood out of the cell down its concentration gradient, now reversing the electric force again. The electric force reemerges, minus 10 millivolts, minus 20, minus 30, minus 40, minus 50, minus 60, minus 70, and about minus 70 millivolts, the potassium channels close. So what we've had here is, first, sodium channels opening, allowing sodium ions to flood into the cell, reversing the electric force at that location in the membrane. Then at about plus 30 millivolts, the sodium channels close and the potassium channels opened up, allowing potassium to flood down its electrochemical gradient to the outside of the cell, bringing the electric force back from plus 30 millivolts to minus 70 millivolts, the resting potential. And then the potassium channels closed. What this means is that the action potential is composed of sodium ions entering the cell, reversing the electric force, followed by potassium ions leaving the cell, restoring the normal resting potential. Here is an animation of the conduction of an action potential. Shown in red is the reversal of the electric force across the membrane. That's the result of sodium ions entering the cell. This is immediately followed by potassium ions leaving the cell, restoring the electric force. The action potential in a neuron is a temporary change in permeability of the axon membrane, first to sodium ions, then to potassium ions, resulting in sodium entering the axon followed by potassium leaving the axon. Sodium in, potassium out. Sodium in, potassium out. Sodium in, potassium out. Sodium in, potassium out. Conducted from one end of the axon to the other. the action potential. Here you're looking at an action potential being conducted along an axon. The red area is the result of sodium ions entering the cell. The blue area that follows the red area is a result of potassium ions leaving the cell. Above, you see a diagram of the two types of channels in the axon membrane. The sodium channels are shown in blue, and the potassium channels are shown in yellow. During the depolarization phase of the action potential, the sodium channels open, allowing sodium ions to move down their electrochemical gradient into the cell. Then, during the repolarization phase of the action potential, the potassium channels open, allowing potassium ions to leave the cell down their electrochemical gradient. Note that the action potential is conducted from one end of the axon to the other, like a row of dominoes knocking each other over. The first domino to fall triggers the second one, which then triggers the third, and so forth. This is called a chain reaction. In an axon, the chain reaction is called an action potential and is conducted along an axon without a loss of amplitude. Here you're viewing the plasma membrane of the axon of a nerve cell. In this membrane, we have two types of ion channels. In green, we have a potassium channel, and in black, we have a sodium channel. Notice that both channels are gated. The gate may be open, or the gate may be closed. When the gates are open, the ions can cross the membrane. When the gates are closed, the ions can't cross. On your right, we have a diagram monitoring the electrical activity at a particular location along the plasma membrane of an axon. We can place one electrode of a voltmeter just inside the plasma membrane and a second electrode just outside the plasma membrane. When no action potential is being conducted along that membrane, the electric force is stable at about minus 70 millivolts. This black line represents the so-called resting potential of a nerve cell axon. But a disturbance in the membrane can cause sodium channels to open, allowing sodium ions to rush into the cell, taking their positive charge with them. This influx of sodium ions causes the depolarization of the membrane from minus 70 millivolts, or resting potential, to about plus 30 millivolts. We say the membrane depolarizes. At the peak of depolarization, the sodium channels close and potassium channels open up, allowing potassium to leave the cell. The movement of potassium ions out of the cell with their positive charge causes the electric force to return to resting potential. We call this the repolarization phase of the action potential. Here's the depolarization phase, where sodium ions enter the cell, followed by the repolarization phase, when potassium ions leave the cell, restoring the cell to resting potential. So the action potential is sodium ions in, followed by potassium ions out. This is how it looks to us as we monitor an action potential passing by a particular location on an axon membrane. We do have a bit of a problem, though. The action potential allows sodium ions into the cell and potassium ions out of the cell. With these ions accumulating on the wrong side of the membrane, how are we going to generate future action potentials? The solution is active transport. In the axon membrane is a pump that moves sodium ions out of the cell and potassium ions into the cell. Because we are moving these ions against their electrochemical gradient, we must expend cellular energy in the form of ATP. We call this pump the sodium-potassium ATPase. It does for the neuron what your alternator does for your car. Your alternator recharges your battery after you use the battery to start your car. Without the sodium-potassium pump, the neuron would lose the capacity to conduct action potentials. Here is the sodium-potassium ATPase in action, pumping 3 sodium ions out of the cell for every 2 potassium ions returned to the cell. This labeled graph of the action potential shows the changes in the electric force across a membrane when an action potential passes by. First, we have the resting potential, which is near minus 70 millivolts in a resting nerve cell axon. A disturbance of the membrane causes sodium channels to open. Sodium rushes into the cell down its electrochemical gradient. As a result, the electric force across the membrane moves from minus 70 millivolts to a peak of about plus 30 millivolts. At that point, the sodium channels close and the potassium channels open up, allowing potassium to leave the cell, returning the electric force to resting potential. When the membrane returns to resting potential, the potassium channels close, and the active transport pump takes over to remove sodium ions from the cell and return potassium ions into the cell. The membrane is now ready to conduct another action potential. Here you're viewing the change in permeability of the axon membrane during an action potential. The action potential is shown in blue. The red line shows the change in permeability to sodium ions. The increased permeability to sodium ions is responsible for the depolarization phase of the action potential. At the peak of the action potential, the sodium channels close and sodium permeability drops to normal. But at that point, the potassium permeability, shown here in green, increases significantly, allowing potassium ions to leave the cell, returning the electric force to resting potential. Then the potassium channels close, returning potassium permeability to the resting state.